Okay, so a pleasant day to all of our participants in this Zoom space. Thank you very much, Charlotte, for that very generous introduction. So our team would like to thank the National Crop Protection Center, headed by Director Ebuenga, the DA, Bureau of Plant Industry, represented by Dr. Yago and Chief Wilma Quaterno, and the Food and Agriculture Organization of the United Nations, headed by Dr. Hadley, for this opportunity to share our experience on the species verification and strain identification of Spodoptera frugiperda, commonly known as the fall armyworm, or FAW, or FAW. So I will be using those names interchangeably during the presentation. So in this presentation, we will share to you a story by answering the following questions. First, how was the FAW's presence in the country verified? Second, what are FAW strains? Third, what are the differences between these FAW strains? And lastly, what are the FAW strains present in the Philippines? The presence of FAW in the Philippines was verified using both morphological and molecular analysis. In 2019, samples from Gonzaga, Cagayan, as well as Lawag, Ilocos Norte were provided to our laboratory by scientist Mario V. Nabasero of the National Crop Protection Center. Easily recognizable larval characters of the fall armyworm, as discussed by Dr. Jess Adorada earlier, was noted by the group of scientists Navasero in these samples. These samples were then subjected to DNA barcoding in our laboratory for species verification. First, I would like to give some background on what DNA barcode is. Presented here is the complete mitochondrial genome or mitogenome of FAW. It has 15,365 base pair and has the typical gene order found in Noctuidae mitogenomes, which includes 13 protein coding genes, 2 ribosomal RNA genes, 22 transfer RNA genes, and an AT-rich region. The mitogenome can provide important information for understanding molecular evolution as well as phylogeny. The commonly used DNA barcode for animals, the Cytochrome C oxidase subunit 1, is found in the mitogenome. It is highly efficient for discriminating vertebrate and invertebrate species and has been proposed as the universal barcode locus. To qualify as a DNA barcode region, a locus should be easily amplified using a set of universal primers and should show low intraspecific but high interspecific divergence. So COA1 satisfies this criteria for most groups, therefore has been used for species identification, taxonomy, and phylogenetics. The COA1 gene can be amplified using 3 sets of universal primer. The 5' region can be amplified using TYJ140 and C1N2087 that yields a 650 nucleotide fragment. The 3' region, on the other hand, can be amplified using C1J2092 and 3014 that yields a 950 nucleotide fragment, while the central region can be amplified using LEPF1 and LEPR1 and yields a 700 nucleotide fragment. The primers used for DNA barcoding to differentiate FAW from other Spodopterans are LEPF1 and LEPR1. On the other hand, Nagoshi specified the COA1A region, which could differentiate the FAW strains. The amplicon size ranges from 682 to 685 nucleotides. Using the samples mentioned earlier, The total genomic DNA was extracted from the larval tissues. So this table summarizes the obtained COA sequences as compared with the nucleotide sequences in the gene bank using the basic local alignment search tool or BLAST-N. All samples had a significant hit of 100% identity with nucleotide sequences of FAW with gene bank account number MK318297.1, except for this SL3 with 100% nucleotide identity with FAW with gene bank account number of MK381422.1. So nonetheless, both of these significant hits are from the study of Gilligan on the identification of heliotine larvae intercepted at the U.S. ports of entry from the New World, which was published in the Journal of Economic Entomology. So our results were also published in 2019 in the Philippine Entomologist with scientist Navasero as the lead author. Should you need an e-copy of this paper, please do not hesitate to contact us. So in the previous slides, the FAW strains were identified through COA nucleotide sequence. So what are these FAW strains? So it was actually Dorothy Pashley who subdivided FAW into two strains on the basis of genetic differentiation associated with larval host plants. One strain feeds primarily on corn, but also feeds on sorghum and cotton, and was called as the corn strain, or CS for short. The rice strain, or RS individuals, primarily fed on rice, but also exist on alfalfa, pasture grasses, and turf or small grasses. So the groupings of corn and rice strains was further confirmed by Pashley in a follow-up study on the loci of esterase, hydroxybutyrate dehydrogenase, and peptidase. So several characters has been identified to differentiate the two strains. For instance, several molecular markers were employed to differentiate the strains. So these are the allosines, the esterases, through PCR RFLP, FR fragment repeats, AFLPs, mitochondrial genes, ND1, ND4, and CO1. And in addition, the genomic sex-linked triose phosphate isomerase, or TPI gene, has also been used to identify the strain of FAW. To date, no morphological difference between the FAW strains has been reported yet, except those reported by Cañas Hoyo and colleagues. Interestingly, they noted a difference in the wing shape and size between these strains. Studies also shown that although these strains are sympatric, they evolve prezygotic and postzygotic isolation mechanisms. For instance, there is a temporal mating isolation as female calling behavior occurs earlier in the corn strain. Behavioral mating isolation is also observed as female corn strain rarely mates with male rice strains. There is also chemical mating isolation as the strains differ in pheromone component ratio. And lastly, hybrids produced in the laboratory exhibit reduced fitness. So these hybrids have a reduced number of egg masses deposited, reduced number of larvae, reduced number of females, shorter pupal development time, and shorter female and male longevity. The strains are also reported to differ in tolerance or susceptibility to insecticides and biocides. For instance, rice strain is more tolerant to lambda cyhalothrin and methomyl than the corn strain, while the corn strain is more tolerant to methylparathione, cypermethrin, and cry1AC and cry1AB proteins of Bacillus thuringiensis than the rice strain. So we're now in the exciting part of the story. So what are the FAW strains present in the Philippines? So we have an ongoing study on the genetic structure of fall armyworm in the Philippines. The following slides now briefly summarize the protocol we have developed in our laboratory. So larval specimens were collected from corn and rice growing areas in the country in collaboration with the National Crop Protection Center, the Bureau of Plant Industry, and the regional crop protection centers. These were preserved and were first morphologically analyzed and photo-documented in the insect taxonomy laboratory of Dr. Jessamine Adorada. So the total DNA of each individual was extracted from the thoracic muscles using a commercial kit with a 5-step protocol. And the extracted DNA was then used as template to amplify the partial COA1 sequence, and the samples with amplicons were visualized and verified in agarose gels. These amplicons were then sent for sequencing to a third-party service provider, and the nucleotide sequences were pre-processed in bioedit and were consequently subjected to identity mining using BLASTn across the gene bank database. Preliminary phylogenetic analysis was done using the neighbor joining method in MegaX. We have collected FAW samples from 15 regions in the country. However, we're still working on some specimen, and therefore we can only share our partial data. So the blue triangle in the map represents the corn strains, while the red circles indicate the rice strains. So the presence of corn and rice strain based on the partial COA1 nucleotide sequences can be observed in Luzon. So rice strain individuals are dominant compared to corn strain. This slide represents the samples gathered from Visayas. Based on our partial analysis, most individuals are rice strain, while only a few are corn strain collected. Lastly, this slide shows some FAW specimens gathered from Mindanao. Likewise, rice strain individuals are more frequently observed than the corn strain. So meanwhile, we also analyzed the TPI sequence of some of our samples. So based on our preliminary analysis, we have not yet found any heterozygote in our FAW collection. So that's our story for now, and we hope you find this very interesting. So we have drafted a manuscript regarding the genetic structure of FAW in the Philippines. So we will inform you once the publication is already out. So on behalf of the team, I would like to acknowledge the following for their invaluable assistance in the collection of the FAW samples. Likewise, the following are acknowledged for their assistance in the collection of our samples that made us proceed with our project even we are in pandemic. We also thank Picard BOST for funding our research. So thank you very much for listening.